Welcome, everybody. I'm going to give you an introduction into RNA-based cancer vaccines, what they are and how they might work. And then I'll hand over to David to talk about real clinical implications and how they're currently being used. So when we think about vaccines, I looked up on Wikipedia, the great source of information as to how people are defining a vaccine. And you can see the definition at the top of the slide there. But essentially, the way we make vaccines has come an enormous way over the first original discovery by Jenner that one could use cowpox, isolated from initially the scabs of cows infected with cowpox, and use that to vaccinate individuals to protect them against smallpox. And you'll know that smallpox has now been eradicated. But essentially, that was a very undefined mixture of virus and other things, not really a very good biological process, where we've now moved from that to being able to use defined genetic sequences. So no longer parts of viruses or whole viruses themselves or whole tumors. We can isolate parts of the genetic information that are required to express a protein that induces an immune response. So this means it's much more controlled and essentially a much safer product because you're having to control much fewer things in order to get that highly purified vaccine. And when I talk about RNA technology, people often feel that it was developed too quickly because the public perception is that it really only came to the forefront of people's attention during the pandemic, as if it was developed over a 2-year period. But actually, it's building on at least 40 years worth of discovery and development. I won't go into that history, but you may be aware that the Nobel Prize was awarded last year to 2 leaders in the field who worked out how to stabilize RNA and be able to use it more safely. So this is not new technology. This is technology honed through years of research and continues to be further developed. Now, when we think about this technology, I'm sure you're all very familiar that DNA makes up your chromosomes. That's what makes you who you are. And the DNA essentially wants to stay as a constant repository of information to make proteins. So ideally, your chromosomes are not altered, except in the case of tumors where you may get an alteration in your chromosomal sequences that lead to altered proteins. RNA is what we call the messenger, and normally it's produced in the nucleus. It carries the code to be translated and make new proteins. RNA is very fragile and degrades very quickly, and that's part of the way cells control the amount of protein is made and for how long it's made. Now, that's both an advantage and a disadvantage for gene delivery. The advantage is it doesn't hang around for very long. It has no possibility of altering your chromosomes, and so it's very short-lived and safe. The disadvantage is we have to work out how to stabilize it and deliver it into cells. And that's part of that discovery process that came about over the last 40 years. Now, this slide looks a bit scary, but this string of letters is essentially the genetic code for one of the mRNA vaccines. It's actually the Pfizer vaccine. And what essentially it says is we are just making a string of nucleic acid, and that's being used to encode the spike glycoprotein, in this instance, for the SARS-CoV-2 virus that causes COVID-19. Now, that can be swapped for a mutated sequence of a protein associated with a tumor. Now, one of the important aspects of understanding how to use RNA was to work out how to deliver it in the body to cells. And in this slide, what you see is this little specialized unit, which is essentially a nanometer-sized lipid droplet. It's a very sophisticated lipid droplet, but it captures the RNA within that droplet, and that protects it from the harsh environment and delivers it into cells. And when it gets into cells, it delivers the RNA into the cytoplasm, and the cells see this as the instructions to make the protein that it is encoding, whether that's a viral protein to induce an immune response against an infectious disease, or a tumor protein to induce a response against the tumor. We work with different types of RNA. This is what we would call conventional RNA. It gets into the cell and it just produces a limited amount of protein. For some indications, we use what we call self-amplifying RNA, which can also amplify the number of copies in cells and express protein for longer periods of time. And in terms of immune response, we use those for different indications. So we know how to manufacture these very highly purified, highly defined genetic sequences and capture in these very specialized lipid droplets. And that's what's injected into your arm. Some of the material drains to the draining lymph node, which is where your immune sensing cells sit, and that elicits an immune response when the foreign protein is made. Now, as you'll see in a moment, that's fairly straightforward for an invading pathogen, because those proteins look very different to your own proteins. And so the body is trained to be sensing and looking for anything that's not you. And if it's not you, it assumes it's an infectious threat and makes an immune response. So how do we take the same technology that was used for coronaviruses and move that into developing vaccines? Well, this isn't something that's new. In fact, the technology, RNA technology, was originally developed specifically for cancer vaccination. And so this research has been going on for quite some time. It's only because of the global success in the infectious disease space that many people have started to realize the potential of the technology. Now, this is a nice slide looking at the immune response. And if you're not familiar with this, essentially what is happening is the RNA is being delivered into the cell. It's making those proteins. And in the context of making responses against tumors, it chops those proteins into small fragments that are then presented by your MHC, your major histocompatibility complex. It's a long word to say, but this alerts what we call killer cells or cytotoxic cells that are then programmed to seek out, in the case of infection, an infected cell, or in the case of a tumor that is expressing a mutated protein. And it allows these cells to kill those tumorous cells, ideally completely getting rid of the tumor, or at least giving very potent levels of suppression of tumor growth. Now, in order to do that, we need to determine in a tumor what is different from your own cells. Because as I've told you, your immune system is really programmed to identify things that are not you and are not normal. And so some tumors have what we call shared neoantigens. And a neoantigen is a mutated protein that is not expressed in normal tissue. So ideally, the best vaccines are one where we can make the same vaccine and use it in multiple individuals against a certain tumor type. Now, while this is the most optimal approach, there are relatively few antigens that are shared across tumors between individuals. So increasingly, and the exciting really potential of this technology is that now we can start to look at what mutated protein is expressed in an individual patient, and then identify that through sequencing to make a vaccine with the genetic code that only encodes that mutated protein and can be delivered to the individual. Now, this approach would not be possible without RNA technology and the speed at which both these antigens can be identified through high throughput sequencing and the ability then to make RNAs specific for that individual. Now, I want to stress this is still very much at the early stages of development. It's not something that you can go to the hospital today and say, I want a personalized vaccine. It will be technology that is increasingly used as time goes by and as we get better at working out exactly what's required to induce those immune responses. This is showing in cartoon form what normally happens in a normal cell. You have your DNA, it makes RNA, it makes a protein, which is essentially made up of amino acids, the building block of proteins, and that folds into the finished protein. And because that is you recognize it as your own protein, you make no immune response. With a mutated cell, it may differ by a single amino acid that is responsible for causing the tumor. And so then this is not the same as a normal protein. And what we need to do is drive an immune response to recognize that small difference. Now, to do that, people take a sample of normal cells and a sample of the tumor and then compare the DNA between both, and also sometimes at the RNA level, to detect a single change in the genetic code, or maybe several changes that can be used to identify what are the parts of the protein or proteins that we need to target. What you can see here is a genetic sequence of a virus is completely different to a human. So this is why this is relatively easy to do. And in a tumor, you're just seeing a single change, single base change in the genetic code. So now with high throughput sequencing and very sophisticated computer technology and increasingly using artificial intelligence, one can start to look at these genetic sequences between tumorous cells and non-tumorous cells and predict the protein that one needs to get the immune system to target. But because the differences between a tumor cell and a healthy cell may differ only by one or a few amino acids, it's really not as easy to evoke an effective immune response as it is for a totally foreign infectious protein. And so often these therapies will be used at the same time as using technology to essentially reduce some of the safety components of the immune system that are trying to protect an immune system for being too overly sensitive. So we use what are called checkpoint inhibitors. Often these are antibodies that essentially allow an immune response to occur to very small differences and induce those more potent T cells to something that only differs by a very minor margin in order to wake up that immune response. Now, the advantage of the RNA technology is that the way to synthesize RNA is really very easy and can be done in a matter of days. And in terms of cancer, time is of the essence. And so being able to identify those differences in the tumor proteins, make the RNA, and then get it back into the individual can be now completed within a meaningful time frame. And then finally, how do these anti-tumor vaccines get used? Well, they are most often used by either injecting direct into the tumor, because often uptake of these vaccines into the tumor can be relatively poor, or through intravenous infusion. There are other ways to use it, but those are the most common ways of delivering a novel vaccine against a tumor. Now, we're very excited about this technology from a scientific point of view, but also in terms of a health point of view, because it can work now against infectious diseases, cancer immunotherapies, which we're talking about today. There are people looking at using it for allergen tolerization, protein replacement, genome editing, and genetic programming. So this is a technology that I think over time will increasingly be used across a wide range of medical disciplines. And I'll stop there. Thank you for your attention.